So thank you all for joining us. My name is Manisha. I'm a program manager at Seven Bridges. I work with the Cancer Genomics Cloud. And I'm very excited to tell you today about some tools that we've developed to make it easier for single-cell researchers to do their data analysis on the Seven Bridges platforms. The reason why single-cell analysis has grown so much in the past several years and been so impactful in transforming many different fields of biology is that understanding the individual pieces that go into a tissue or a system is hugely important to understanding the properties that emerge from that tissue. So looking at, for example, cancer stem cells can give you a much better sense of how cancer progresses. And by looking at things individually, you can get greater insights from the smaller populations that are likely driving the true biological changes, like, again, for example, cancer stem cells. And you can also use these insights to identify potential marker genes for therapeutic targets or biomarkers. However, as the pace of single-cell research has exploded, there continue to be lots of challenges with them. One of the primary challenges is, as I mentioned earlier, the data sets can be huge and very complex. So millions of data points, each of which is a whole transcriptome, a whole genome, et cetera. And this can make it really difficult to access and compute on these data sets. And that's where the Seven Bridges can really help a lot by keeping the data in the cloud and the analyses in the cloud. We make it really easy for anybody to be able to access and compute on these large data sets. And then while these data sets are very large and complex, the actual individual data from a single cell is not that huge. So they're actually, the data itself is actually quite sparse compared to a bulk RNA-seq experiment, for example. So this means new methods for looking at this type of data have to be developed. And in the past 5 to 10 years, there's been a huge number of tools developed. You can see on the graph to the right from an organization that keeps track of these tools, there's over 600 tools now available for doing single-cell analysis. And there hasn't quite been a consensus yet in the field on which tools to use. So it can be quite confusing for somebody new to the field to know exactly which tool to use and how to set it up. And this is, again, where the platform can be very helpful. We have publicly accessible apps, so you don't have to build the tool yourself. They're pre-built on the platform for you. And what we're going to do today, actually, is talk through some of the most popular tools that we've added to the platform and do some comparisons between them to make it a little bit more clear which tool might be most helpful for your research needs. And you can see the growth in the field by the growth in single-cell atlases that have come out in the past few years. So the first set of atlases that came out, not surprisingly, were from very simple organisms like C. elegans and fruit flies. But more recently, more complex mammalian atlases have come out. There are multiple ones now of the entire mouse brain, the whole mouse nervous system. There are atlases out for every tissue within mouse. And there are many human projects that are in progress, which are extremely complex. So the Human Cell Atlas is aiming to characterize every healthy tissue in the body. And the Human Tumor Atlas Network, for example, is looking at single-cell characterization of tumors. And you can see an example of one of these on the right here. It's looking at zebrafish developmental biology, and they were able to basically map at a single cell level every cell in the organism and identify which cell types were present in there. And they did that using the Seurat package, which we will talk about more in the rest of the talk. And not only that, they were able to look at lineage by reconstructing the developmental trajectories. So not only do they know what cell types are present, but they know how they're connected and in what order they developed by making these single cell maps. So this gives you a much more comprehensive picture of what's happening in the biological development of these organisms. And we've also made tools to do these trajectory analyses available on the platform as well. And we'll actually talk about this in the next section as well. So now I'm going to transition over to Nemanja, who will go through the actual tools themselves.
Thank you, Manisha. We can start off with the next slide, please. Okay, so today we will be talking about tools and workflows optimized for processing both UMI-based and full-length single-cell protocols We will be presenting R packages that could be used for downstream single-cell analysis performed on gene-to-cell counts. Also, we will present the results of comparative benchmarking of different tools for processing 10x Chromium datasets in cloud-based environment. And we will talk about how we analyzed full-length data to understand changes in the endothelial tumor cells over time. Next slide, please. Okay, we'll start off with presenting tools and workflows optimized for processing both UMI-based and full-length single-cell protocols. Next slide. One feature of single-cell analysis is the existence of a large number of sequencing protocols. The main difference between protocols is whether they capture the full length or just one end of a transcript. Full-length protocols developed for single cells are quite similar to protocols for bulk RNA-seq. They have lower multiplexability, meaning that you can combine fewer cells per sequencing run. The number of cells that can be sequenced highly depends on the desired sequencing depth. The full-length protocols are useful for cases where higher sequencing depth is required, such as identification of single nucleotide variants, mutations, RNA splice variants, or T-cell receptor sequencing, which is the example shown on the right. On the other hand, protocols that sequence just the ends of transcripts have introduced barcode and UMI labeling to one end of the transcript. Cell barcodes enable labeling of each cell and expands multiplexing ability to more than 1,000 cells per sequencing run. The use of UMI sequences will be explained later. Combining more cells means that the sequencing depth of these protocols are usually lower in comparison with full-length protocols, and they are used for dissecting heterogeneity of complex tissues and identification of marker genes, which could lead to detection of novel cell types. Next slide, please. So tools for processing full-length single-cell protocols are pretty much the same ones used for processing bulk RNA-seq, but optimized for single-cell data. Data produced with full-length protocols may come already demultiplexed, where each pair of FASTQ files carries row sequencing reads of a single cell. If that is not the case, demultiplication is the required step in the analysis. Some of the splice-aware aligners, such as STAR or HiC2, can be used for aligning reads to the reference, and tools like RSAM or PH.D can be used for quantification, producing tables of read counts per gene or per transcript as a result. There are many extensions of tools used for bulk RNA-seq that are optimized for processing single-cell data produced with UMI-based protocols, and we will be mentioning some of these in more detail later on, but the main addition to these tools are the barcode demultiplexing and UMI deduplication steps that are mandatory for UMI protocols. Next slide, please. Okay, so cell barcodes refer to the unique sequences added to each fragment that came from a single cell before all cells are mixed together and sequenced. UMI-based protocols use additional type of barcodes called UMIs. These are unique molecular identifiers that are short random sequences added to the transcript during reverse transcription. They enable sequencing reads to be assigned to individual transcript molecules and thus enable the removal of amplification noise and biases from single-cell data, which provides a great improvement in quantification step of the analysis. UMI sequencing typically produces paired end reads, where one read from each pair captures the cell barcode and UMI, while the other read consists of exonic sequence of the transcript. Next slide, please.
Okay.
Okay, so cell barcode demultiplexing refers to the step in processing where the cell barcode information is used in order to know which sequences came from which cells after they had all been sequenced together. So the sequences with the same barcodes are grouped together. The UMI deduplication step removes the duplicate read sequences that came from the same transcript. One of the main challenges with analyzing this type of data is dealing with sequencing errors present in barcode and UMI sequences, and different single-cell tools for processing this kind of data have different approaches that can be used for processing UMI-based single-cell protocols. And tools are separated into two groups based on alignment approach they use. So on the left are the alignment-based tools such as Cell Ranger, Zoomis, and Star Solo that use splice-aware algorithms for aligning read to the reference that is already implemented in Star Aligner. On the other end, there are extensions of Callisto and Salmon, and these are the tools that use pseudo-alignment algorithms for single-step alignment and quantification that is optimized for single-cell data. In the middle are UMI-based single-cell protocols that are supported by each of these tools. Tools are connected to the protocols they support with lines. And all of the tools support the 10x Chromium protocol, and some of the tools such as Zoomis, Star Solo, and Salmon have option to arbitrarily set the start and end positions of barcode and UMI sequences in second read, thus enabling them to process data produced with practically every single-cell UMI protocol. Next slide, please.
Okay.
Okay, so we use a common workflow language to describe software packages for processing single-cell data and make them available on Seven Bridges cloud environment in a form of tools, workflows, or as interactive analysis. Tools would represent a single method for one step of analysis, while a series of tools connected together into a pipeline represent workflow. Interactive analysis on Data Cruncher are Jupyter or R Markdown notebooks, providing higher-level analysis of processed data that includes visualizations and other interactive components. Next slide, please.
Okay.
So to summarize the first part of our talk, here is an overview of all single-cell tools we have available for both full-length processing, UMI-based processing, and higher-level analysis using R packages. Again, the goal of the processing step is to go from raw sequencing reads to some form of counts data. And for processing full-length single-cell data, we have a SmartSeq2 workflow, which we will describe in more details in the last part of the presentation, and two tools, Tracer and MixCR, that can be used for the reconstruction of T and B cell receptors. For processing UMI-based single-cell data, we created a Callisto bus tools workflow. We also have a Cell Ranger toolkit consisting of 5 separate tools. And we have extensions of Star Aligner, that is called Star Solo, and Salmon Pseudo Aligner, which had extension called Alevine. In the Data Cruncher section, which we will discuss more in the second part of the talk, we have a single-cell interactive analysis, and this analysis is executed within RStudio environment on Seven Bridges Platform. And it includes notebooks that, as an input, takes count data and perform additional analysis and visualizations. We have 3 separate analyses created as R Markdown notebooks, which can be used to perform correction of patch effect using Harmony R package, clustering and detection of marker genes that is based on Seurat package, and trajectory inference based on Monocle. All of these notebooks use the results that are produced with tools and workflows that we already mentioned. Next slide, please.
As we get into the actual tool usage, I just want to mention that if you have questions, please type them into the chat window and we'll try to address as many as we can at the end.
Okay. So in the following section, we will be talking more about analysis available on Data Cruncher. that can be used to perform downstream processing of gene-to-cell counts. Next slide, please. So single-cell downstream analysis starts with a gene-to-cell count table produced as a result of quantification using the tools described in the previous section. And these tables hold information about the number of reads or UMIs that are assigned to each gene and each cell. Rows of the table representing names of genes, while columns representing different cells. We'll give a brief overview of some downstream analysis that could be performed on gene-to-cell counts. Next slide, please. Okay, we'll start off with the correction of patch effect. So patch effect is usually present when merging single-cell data generated at different times or different laboratories, which can introduce technical variability. So the purpose of patch correction analysis is to remove the technical variability and preserve biological variability in dataset. The best display of patch effect is visual representation of datasets reduced to 2 dimensions using t-SNE. And in the top right image, we compare cells from 2 datasets, one colored blue and the other one colored orange. So the blue and orange are clearly separated on the top picture, and this is an indication of patch effect present after merging 2 datasets. After identification of the patch effect, we can remove it using the Harmony R package. On the bottom picture, which is the same data processed with the Harmony package, we observe no clear separation between cells originating from different datasets, thus confirming we have successfully removed the patch effect. Next slide, please. So the second analysis we would like to highlight is clustering and detection of marker genes that is based on Seurat R package. Seurat analysis consists of several steps, which include selection and filtration of cells based on some QC metrics, data normalization and scaling, detection of highly variable genes, dimensionality reduction using PCI, clustering of cells, visual representation of cell clusters in 2 dimensions, and detection of marker genes within cell clusters. So on the image to the right is a t-SNE plot showing the results of clustering in 2 dimensions, and we observe 8 clusters representing distinctive cell types. And here cell types were detected and annotated by analyzing marker genes that are expressed in each of these clusters. Next slide, please.
Okay.
So the third package we'd like to highlight is Monocle. And cells that exist in different stages of development within the same population can display different transcription profiles over some time period. The package Monocle provides methods for ordering single cells and placing them along a trajectory that corresponds to a biological process such as cell differentiation. In the first step of this analysis, it is important to remove empty cells and doublets, and those would be the places where 2 or more cells are sequenced by accident. And Monocle then employs a differential expression test to reduce the number of genes. This step requires prior knowledge about cell types and genes expressed in those cell types. The alternative is to perform unsupervised clustering before this step. In the next step, Monocle performs dimensionality reduction, followed by construction of minimum spanning tree. And in the final steps, Monocle algorithm finds the longest path through the minimum spanning tree, corresponding to the longest sequence of transcriptionally similar cells. We'll be talking about trajectory inference more in the final section of the presentation. Next slide, please. Okay, so now that we have gone through the tools and packages available for single-cell analysis, we will present some examples and demonstrations of how they perform. And first, we'll present the results of comparative benchmarking of different tools for processing 10x Chromium datasets. Next slide, please. Okay, so we did performance benchmarking to compare the execution runtime and AWS costs of 4 single-cell processing tools on 10x genomics datasets. So left plot represents the relation between total size of 10x dataset and execution time on Seven Bridges platform. And on the right plot, you can see the relation between the dataset size and AWS costs. We observed that pseudo-alignment-based tools such as Salmon and Callisto, and that would be the ones in the plot that are colored in blue and red, and those have significantly better performance in terms of execution time and AWS cost in comparison to alignment-based tools such as CellRanger and Zoomis. On the following slides, we will talk more about methods we use to compare the precision of the obtained results. So we compare the accuracy of the results produced with different tools on multiple levels of single-cell analysis. For the quantification level comparison, we simulated a single-cell dataset and also used a bulk RNA-seq data as a second approach. We also performed clustering and gene marker comparisons using interactive analysis based on Seurat package. And for this purpose, we use 2 datasets that are labeled as gold standard and silver standard. And this gold standard dataset is a low complexity dataset created as a mixture of 3 human cell lines that are mixed in equal proportions. And the silver standard dataset is a real complexity dataset. It is representing peripheral blood mononuclear cells, and this dataset is downloaded from 10x Genomics website. So cell type labels for each cell and marker genes for distinctive cell types were available, and we used them as a ground truth in our comparisons. And since the ground truth information related to this dataset is obtained using a different approach that is less reliable than used for gold standard dataset, this one is labeled as a silver standard. So we'll present the results for the silver standard dataset because they are more divergent and provide better insight when estimating precision of these tools. Next slide, please.
Okay.
So we processed simulated FASTQ files with 4 different tools and used gene-to-cell counts as a ground truth for comparing precision of quantification results. Prior to comparisons, genes were split in 10 groups based on the sequence uniqueness. Salmon, Callisto, and CellRanger tools showed high correlations to the truth datasets for the highly unique genes, while Callisto had the highest correlation in the groups of moderately unique genes. On the other hand, Zoomis showed the lowest correlation to the truth dataset in comparison to other tools, and based on these results, we can say that Callisto on this test performs best. Next slide, please.
Okay.
So here we compare the results of clustering performed on silver standard dataset, and we used adjusted rand index and hit homogeneity score for validating the results of clustering given ground truth. And both scores will have a value close to 0 for random labeling and exactly 1 when the cluster labels are identical to truth. So here we purposely enforced the identification of 11 cell clusters because this many reference subpopulations was reported in truth dataset. Also, we performed analysis with Seurat's default settings. From the table, we observe Salmon and Callisto had slightly better adjusted rand index score and homogeneity score than the CellRanger, while Zoomis had the lowest scores in comparison to other tools. Again, Callisto seems to be performing the best in comparison to others. Next slide, please.
Okay.
Here we use Seurat to identify marker genes expressed in distinctive cell clusters in a silver standard dataset. And based on identification of marker genes, all of the tools were able to identify the majority of cell types, But again, Callisto performed best, enabling detection of 8 out of 11 different cell populations. On the table below, you can see that plus sign means that cell type was identified. And we observed that no tool was able to identify all cell types that exist in dataset. But nevertheless, Callisto was able to identify one more than the others. Okay, next slide, please.
Okay.
So our benchmarking results indicate that pseudo-alignment-based tools are much faster and require far less computational power than tools based on star aligner when processing 10x single-cell datasets. Pseudo-alignment-based tools give accurate results and in some segments outperform star-based tools, which we have witnessed on multiple steps when making comparisons. But nevertheless, we advise that Cell Ranger software be used on 10x data since it is optimized and extensively tested and validated. Additionally, Cell Ranger software provides ability to perform clustering and detection of marker genes without the use of downstream analysis based on Seurat. And the final remark would be to use pseudo-aligners rather than zoomies as an alternative to Cell Ranger when working with 10x data. Okay, next slide, please.
Okay.
This would be our last example of how these tools can be used, and we would like to present the trajectory inference analysis conducted on full-length data from endothelial tumor cells that are isolated from mice with melanoma. Next slide, please.
Okay.
So to demonstrate performance of the tools for processing full-length protocols and packages for trajectory inference, we perform some additional analysis using a dataset that is available on the Human Cell Atlas data portal. And this dataset comes from a study on tumor development in mice that is conducted in Teichmann laboratory. And the authors performed single-cell RNA sequencing to analyze changes in stromal cells, such as endothelial cells, fibroblasts, and immune cells at different stages of tumor development. They were tracking these changes in mice to whom they injected melanoma cells, and they were collecting samples after 5, 8, and 11 days after injection. And the main focus of the analysis was related to changes in development of T cells and fibroblasts. Therefore, our idea was to expand this analysis to additional cell types by investigating changes in endothelial tumor cells. Next slide, please.
Okay.
So the authors used SmartSeq2 full-length protocol to produce raw sequencing reads. Reads in FASTQ format are available on data portal of Human Cell Atlas, which we recently made available on Cancer Genomics Cloud platform. And to process files, FASTQ files, we created an implementation of SmartSeq2 workflow in common workflow language, and we optimize it to be executed on Seven Bridges cloud infrastructure. The original pipeline is created by the data coordination platform of Human Cell Atlas, and it uses HiSet2 for the alignment and RSEM tool for quantification. And the entire data set contains more than 12,000 paired and FASTQ files, but using the metadata associated with files, we were able to select just the files representing endothelial tumor cells. We processed this subset of endothelial tumor cells, which had more than 1,000 files, paired and FASTQ files, and total processing time on Cancer Genomics Cloud platform was 49 minutes. The analysis was running on 10 parallel AWS instances, each having 36 CPUs and 72 gigabytes of random access memory with total price of $5.5. Next slide, please.
Okay.
After processing raw sequencing reads and producing gene-to-cell counts, we performed trajectory inference analysis using two different approaches. So both of these approaches are using Monocle R package, but the first approach incorporates timescale information where we identify differentially expressed genes between cells from different stages of tumor development. So before composing the trajectory, we filter out empty cells and doublets. We start analysis with Monocle by selecting the genes that are differentially expressed between different times in development. We use these genes in the later steps to perform dimensionality reduction, followed by the construction of a minimum spanning tree. In the final step of the analysis, the Monocle algorithm maps each cell to the trajectory and finds the longest path through the minimum spanning tree, which is the line connecting the largest number of cells. On the top right plot, we show a trajectory along which Monocle placed individual cells, and selecting differentially expressed genes at the beginning of the analysis, we enforce this trajectory to be in alignment with changes in endothelial cells over time. Now that we calculated the trajectory, the additional step in the analysis could be identification of genes that change along this trajectory. On the bottom right plot, you can see 3 genes coding for transcriptional cofactors and changes in their activity over time. You may notice that these genes were not active in early stages of tumor development, but their activity increased in later stages. So this approach could potentially be used for the identification of genes as a potential biomarkers indicating progression of tumor through different stages of development. Next slide, please. And here we would like to demonstrate another, a more comprehensive method when analyzing trajectories of tumors. So in the second approach, instead of selecting differentially expressed genes over time, we use all genes that pass some filtering thresholds. And we use those genes to perform clustering. Then we order the cells based on the results of clustering, and the later steps in constructing the trajectory are the same as in previous example. As a result, you can see that on the top right plot, the constructed trajectory has more branching points, and the cells from different stages of development are more uniformly distributed along the trajectory. So this approach more realistically represents the development process in tumor tissue, where you will have one trajectory along which the majority of cells are aligned, but also many branches which could represent cells that carry some clonal mutations. On the bottom plot, you can see that Monocle splits this trajectory into states. So the additional analysis could be the identification of marker genes for each state that could represent a potential clonal expansion. Additionally, you could use the information about times when cells were collected to specifically track down cells that are at the origin of clonal expansions. So thus this second approach could provide a significant information about cells that are drivers of somatic mutation in tumor development, and use it to create therapy that will most effectively target tumor tissue. Okay, next slide, please. So I would like to finish this presentation with a couple of ideas for application of single-cell sequencing and how the Seven Bridges platform could be facilitated for the future clinical trials. So we mentioned the Human Cell Atlas project that aims to characterize every cell type with the focus on healthy individuals. And this information is crucial for understanding how disease states are different from healthy tissues. One example would be recent finding that the majority of CFTR transcripts in lungs are coming from a novel cell type that was not identified before. So CFTR gene, when mutated, is the main cause of a disease called cystic fibrosis. But now knowing which cell populations are most involved in producing these disease-related transcripts could help scientists create targeted therapy approaches. Another application of single-cell sequencing would be to monitor disease progression and response to therapy, which is highly utilized in the area of cancer research. You can perform trajectory inference to track down which clones or cells are eliminated by chemotherapy and which ones persist or become resistant over time. Also, single-cell transcriptomics gives potential for mapping patients to the right cancer treatment. And additionally, how well a drug works also depends on what is happening in the surrounding tissue. Single-cell analysis of tumor environments provides a complete picture of disease progression and patient's response to treatment. And we showed a segment of such analysis in our presentation today. So all of these and many other questions can be answered using single-cell transcriptomics in combination with bioinformatic tools we presented today, and cloud-oriented infrastructures like Seven Bridges Platform could help manage increasing sizes of single-cell data sets and provide the environment to conduct reproducible bioinformatic analysis.